Okay, hello everyone. Hope everybody had a good weekend and so far a good week. Today I'm going to be talking about Mycobacteria, or the AFB, the acid-fast bacilli. What I did was I combined lecture 1 and lecture 2, and then after I talk about the AFBs, then I'm going to talk about the gram-positive rods. Okay, so are there any questions? I sent somewhat of a review, kind of to give you an idea of what's going to be on the test. Those questions that I sent you, excuse me, those bullets that I sent you are actually the categories of the matching. So when I do the review tomorrow, I'll give you examples of each of those categories. So we'll do the review tomorrow, and then of course we'll have the exam on Friday. And then the lab, again, supplies didn't come in. Our supplies didn't come in, and we're going to try to do the AFB stains, staining tomorrow. And then hopefully you can see what an actual positive looks like. I didn't have any actual, I'm not going to use actual specimens with actual TB, but I'm going to be using control slides. So I ordered control slides, and we're going to be doing hopefully the hot stain and the cold stain. You'll find out about what each of those stains are throughout this lecture. Okay, so let's go ahead and get started. Mycobacteria, or the acid-fast bacilli. Taxonomy, order, family, genus. I'm not going to ask you about taxonomy. These are the various species. Mycobacterium tuberculosis, for short, we call it MTB. M. bovis, BCG. The BCG stands for Bacille Calmette et Guerin. That's actually the name of a vaccine that's given for tuberculosis. And in certain countries, that vaccine is given. But unfortunately, when they give it, the BCG, your PPD is positive. PPD is the test for tuberculosis exposure. But when you get the BCG vaccine, you're going to be practically positive for the rest of your life. So that's one of the questions that they would ask. Have you ever gotten the BCG vaccine? And that's a reason why you're positive. The other reason why you're positive is because you can pick up the bacteria just walking through the mall. Okay, it doesn't take much to get you to seroconvert to become positive. Then M. kansasii and M. leprae, which is the organism that causes leprosy. Then avium complex, Graphulaceum, fortuitum, and marinum. I'm going to be talking about all of these. These are all the testable species of Mycobacteria. All right. So that picture there is a positive for acid-fast bacilli. The easy thing about reading an AFB smear is if you see red, then you need to zoom in on it and make sure that it's not artifact. But chances are, if you see red, because of the high concentration of acid that's used in the staining procedure, you're not going to see too many contaminants or debris, because you're using concentrated hydrochloric acid. So it's carbofusion, acid alcohol instead of acetone alcohol, and then you counterstain it with methylene blue, and it's a pretty easy test. Like I said, there are two procedures: the hot stain and the cold stain, and I'll talk about that later. So these organisms are slightly curved or straight rods, sometimes branching filaments or mycelium-like growth. They're aerobic, and they don't form spores, and they don't have capsules. So acid-fast means that it's able to take up the stain, even with high concentration of acid in the stain. So its ability to keep the carbofusion stain and then counterstaining with the methylene blue. But then, like I said, it has a decolorizer, which includes hydrochloric acid. And The acid-fast bacilli are the only ones that will cause it to turn red. These organisms are non-motile, not easily gram-stained, and they're considered gram-positive, but nobody would think to stain tuberculosis or Kansassia or M. leprae or anything. These organisms have to be stained acid-fast because of the high lipid content in their cells and cell walls. That's why it's we have to use a special stain. There are two types of Mycobacterium: the rapidly growing species, the ones that grow quickly, and it's usually 2 to 3 days, and the rule is less than 7. Those are the ones that are not disease causing. And then the ones that require 2 to 6 weeks to grow, those are the ones that cause disease associated species. So the rapidly growing species are the non-pathogenic, and the ones that take 2 to 6 weeks on complex media usually is the Lowenstein-Jensen or the Middlebrook. Those comes in two tubes. 2 to 6 weeks. If it's positive, then that's the one that's we want to focus on. Incubation is in the standard 35 degrees or 37 with 10% CO2 and in the dark. So it's kind of like, you put them in the incubator and there's no light, right? You close the door, the light turns off. So it's going to be in the dark, incubated in the dark, CO2 incubator. Acid fastness refers to the cell walls resist decolorization by acidified alcohol. Okay, so it resists decolorization by the acidified alcohol. Complex, that's another term, describes 2 or more species, and distinction between species has no medical importance. So a complex is a complex, but just because if you see positive AFB or acid-fast bacilli, that's enough for the doctor to start treatment. Okay. So distinction between the species has no medical importance. The MTB complex, the Mycobacterium tuberculosis complex, includes these species of organisms: tuberculosis, bovis, which is the one used for making that vaccine, which is the bacille calmet at Garin, Africanum, Microti, and Canetti. If you're positive for Mycobacterium tuberculosis, that's reportable. That's reportable to public health. They need to track positive patients for tuberculosis. All right, the Runyon classification system. There's 4 groups. Okay, there's 4 groups, just like the UN groups, but the Runyon classification for non-TB Mycobacterium. NTM stands for non-tuberculosis Mycobacterium. MOTT, MOTT, stands for Mycobacterium other than tuberculosis. So these are the non-tuberculosis species of Mycobacterium. First group is called the photochromogens. First group is photochromogens, and these organisms will grow greater than 7 days. And the characteristic of this group is that the colonies will turn yellow when exposed to light after growing in the dark. Okay, so it's grown in the dark. Once you open the door, you expose it to light, then it turns yellow. Okay, it turns yellow only when exposed to light. And it takes more than 7 days to grow, like I said, so it's a slow grower. And the 2 species for the photochromogens are Kansasii and marinum. Okay, so it turns yellow when you open the door and it's exposed to light. So when the door is closed, it's not yellow, just so that you understand. When the door is closed, it's not yellow. Once you open the door, then it turns yellow. Those are the photochromogens, and they grow greater than 7 days. The next group is the scotochromogens. Now in this group, it'll turn yellow, but it'll turn yellow regardless of whether or not it's in the light or dark. So It's already in the dark when you put it in the incubator, so it's already yellow to start with. With the photochromogens, the first one, it's in the dark, but it's not yet yellow, okay? So it's not yellow, open the door, turns yellow. This one, in the dark, it's already yellow. And then once you open the door, of course, it's gonna be yellow as well. This one also takes more than 7 days to appear on solid media. And the two species in this group are M. gordoni and M. Mycobacterium scrofulaceum. Gordoni and scrofulaceum. The third is the non-photochromogens, okay? So regardless, light or dark, there's no pigment at all. It's non-photochromogen, so there's no pigment, okay? In the dark, not yellow, open the door, it's still not yellow, okay? This also takes more than 7 days to appear on solid medium, and there are two species that Mycobacterium avium complex is actually a complex of several organisms because it's a complex, and then Mycobacterium ulcerans. So these are the non-photochromogens, will not turn yellow regardless of dark or light. Okay, so that's group 3. Greater than 7 days to appear. So the first 3 groups, it takes more than a week before growth appears. The fourth group, group 4, are the rapid growers. These organisms will take less than a week to show up on solid medium, okay? Fortunatum, chelonis, smegmatis, and abscessus. Okay, so the main characteristic is that they take less than 7 days to grow. No information on, I don't know what the information is on color, but if it shows up less than 7 weeks, regardless of color, it's a group 4 rapid grower. Okay, all the other ones, groups 1 through 3, take more than 7 days to show up on solid medium. And then there's a non-cultivatable, tuberculosis Mycobacterium, NTM. So remember, NTM is non-tuberculosis Mycobacterium or Mycobacteria other than tuberculosis. And that's M. leprae, which is leprosy. Okay, make sure you know this because this is a unique, unusual piece of information that is testable. Mycobacterium leprae, which causes leprosy, you cannot grow it on laboratory media like the Lowenstein-Jensen or the Middlebrook. It won't grow on conventional Mycobacteria media in the laboratory. The only way you can grow this organism is using live animals, either on athymic nude mouse or armadillo. The most common test question is which organism which Mycobacterium species will grow on armadillos? And the answer is Mycobacterium leprae, okay, armadillos. Make sure you know that. It's a very testable piece of information. So the two medias that I was talking about are the agar-based medium, which is the Middlebrook. I'm not going to require you to know the 7H10 or the 7H11. It's either the Middlebrook or the Lowenstein-Jensen, Middlebrook or LJ, okay? The egg-based media is green, and the agar-based media looks like an agar slant. Okay, and it's Middlebrook and Lowenstein-Jensen. Those are the solid media for growing these organisms. And then there's liquid media like Middlebrook broth, Bactec broth, Septacheck, Mycobacterium growth indicator tube, or the midget Mycobacterium growth indicator tube. These are liquid media, not commonly used. The common way to culture AFBs are usually the media-based, agar-based, the Middlebrook and the Lowenstein-Jensen, and these are kept and read for 8 weeks, 2 months. Okay, the rapid methods for diagnosis is also available, which is the acid-fast smears. So you take a specimen like a sputum, you'll make a direct smear, or fluid, like a synovial fluid or bronch washing or whatever. You make a direct smear, and they'll take that direct smear and do an acid-fast and AFB stain on it. And if it's positive, then you know right away that the specimen contains acid-fast bacilli. Okay. If it's negative, then they'll go through the digestion and decontamination procedure where you take a sediment and then you make more smears. Okay, so acid-fast smears is a rapid way to identify a GL, a gas liquid, and HPLC, high performance liquid chromatography, is another way to detect the organism, and AFB fluorochrome. Other detection systems, again, BACTEC puts out a lot of different projects like the 4460, the 9000, the BACTEC Midget, ESB culture system too. Not obviously no red, so I'm not going to test you on these. The growth rate, conventional ID techniques. Remember the Runyon groups 1 through 3 grow greater than 7 days, and 4 is a rapid grower, grows less than 7 days. And also 2, look at the pigmentation, the photochromogens, the scotochromogens, or the non-pigmented organisms. Niacin accumulation is another characteristic of Mycobacterium tuberculosis. Niacin accumulation, which looks like breadcrumbs, colonies of breadcrumbs. If you see that on any of your media, then that's a pretty good sign that you probably have Mycobacterium tuberculosis in your culture. Other tests that are useful are nitrate reduction, urease activity, tellurite reduction, and DNA hybridization. The first 3 I'm going to be talking about, and they can be testable information: tellurite, urease, and nitrate. Okay, the Mycobacterium that are clinically significant now I'm going to talk about. And the first one is MTB, Mycobacterium tuberculosis. The picture up here, that's what the breadcrumbs look like. So okay, it looks like greenish media. So that's probably the Lowenstein Jensen. Okay, and then you get this breadcrumb appearance of the colony. That's niacin. Okay, that's a niacin test positive, and it's the single best test to identify Mycobacterium tuberculosis. Then there's a nitrate reduction. The organism resembles breadcrumbs. Okay, the description is breadcrumbs, rough, dry colonies, luxurious growth. It likes 37 degrees, and it's also urease positive. But the 2 main lab tests for MTB is niacin positive, because no other Mycobacterium is niacin positive. That's characteristic for MTB. And then there's a nitrate reduction, and then the urease positive. Tuberculosis, symptoms of this disease is low-grade fever, night sweats, fatigue, anorexia, weight loss. Spreads to the kidneys, spleen, bone marrow, central nervous system, and the GI tract. So the symptoms, those are usually the symptoms that they ask you when they're looking for exposure. You know, have you lost unexpected weight? You experience night sweats, fatigue, anorexia, and weight loss. So those are typical questions that they would ask on a survey for tuberculosis. Associated diseases, this organism is highly contagious. Like I said, you can just by walking through the mall and catching, and it's called droplet nuclei. When somebody coughs, you have droplet nuclei that's in the air. You can actually contract that, and then once you get it into your system, it only takes a few bacilli. Few viable organisms can lead to infection, and then what will happen is you may not get sick, but it'll be enough for you to seroconvert, meaning that when you take the PPD test, you do it for several years and you're positive, you're negative on the PPD. Now all of a sudden, because those few organisms got into your system, now you're going to seroconvert to being PPD positive. Okay, it doesn't take many organisms, but that's how you get it. Droplet nuclei that are inhaled. Mycobacterium tuberculosis has a high prevalence in patients with AIDS. Okay, very opportunistic, very contagious. Associated diseases, causes lung infections. This is a respiratory infection, granuloma, caseous necrosis. Caseous means cheese-like. It actually turns your lung tissue into like a cheese-like consistency, and that's what caseous necrosis is. Ultimately, cavitary disease. All right, and that's a lung that's infected with MTB. If you look at the upper 2/3, a person may contract pulmonary tuberculosis from inhaling droplets from a cough or sneeze by an infected person. So it doesn't, like I said, it doesn't take much to become infected with tuberculosis. Normal lung on the left and infected lung on the right for comparison. Okay, more pictures of lungs. We'll just leave these pictures for the pathologists. And here, radiology, X-ray. Right here is a lesion of a lung infected with tuberculosis, and this one is negative. Okay. Pulmonary tuberculosis, symptoms again, fever, chills, myalgia. Collection of specimens. The best specimens are, and it's combined days. That's why it says it's combined days. First 3 morning sputum specimens, and you can combine them. Like day 1, day 2, day 3. The doctor will give you 3 cups. Day 1, he'll say cough up 3 times. Day 2, cough up 3 times. And day 3, cough up 3 times. And once after you do your 3rd specimen, you got to bring it to the laboratory within 30 minutes. So you want to get a good representation, and you want to capture the organism while it's in your lungs. So those are sputum specimens. Mycobacterium bovis, the next organism, is niacin test negative. Remember, if you get niacin test positive, it's probably MTB, okay? And bovis is niacin and nitrate negative. Bovis is bovine and human tuberculosis. You get it from ingesting contaminated milk, and it's not prevalent, not prevalent due to pasteurization. Airborne, human to human, or human to animal, and animal to human. So this, just like MTB, it is an airborne organism. The next one is Mycobacterium kansasii. Again, niacin test negative, nitrate positive, urease positive, and catalase positive at 68 degrees. But it's niacin test negative. Remember, reserve the niacin test for Mycobacterium tuberculosis. Kansasii has another name. It's called the yellow bacillus. Okay, probably because of the yellow colonies that it grows from incubation. Natural reservoirs, tap water. Associated disease, chronic pulmonary disease, extrapulmonary disease, cervical lymphadenitis, and cutaneous disease. The next one is Mycobacterium marinum, and you get this from the sources freshwater and or seawater. And the people who usually get Mycobacterium marinum are fish handlers or people who maintain swimming pools, or you can be swimming the organisms in there. So again, swimming pools, even though it has the chlorine, it's a very hardy organism because it's hard to kill. So again, because of the high lipid content in the cell wall, and it causes cutaneous infection. So just by swimming in the water or freshwater or seawater, handling fish and swimming pools, you can get it on your skin. That's Mycobacterium marinum. M. smegmatis is normal flora of the genitalia, and it's usually a contaminant. That's Mycobacterium smegmatis. And Gordoni, Mycobacterium gordoni, is also found as a tap water contaminant. The complex, MAC, Mycobacterium avium complex. It's commonly isolated species in the US, especially with HIV patients. It's opportunistic, like I said, ubiquitous, meaning that it's found everywhere, and you can get it either by inhalation or consuming it through your mouth. So that's Mycobacterium avium. Intracellular, actually those two is another complex. It's called Mycobacterium avium-intracellular. And then you have paratuberculosis and the leprae murium. That's not leprae, but leprae murium. Associated disease of this group of mycobacteria is disseminated disease in AIDS patients. So I mentioned AIDS a few times. Again, these patients, if they're immunocompressed, they have a high likelihood of being positive for AFB, okay? Other patients are with pulmonary infections, cervical lymphadenitis, and disseminated diseases. Mycobacterium scrofulaceum, normal habitat is raw milk, soil, water, or dairy products. And the diseases can be bacteremia, cervical adenitis, pulmonary diseases, and skin infections. Leprae, leprosy, no growth in vitro, like I said. It will only grow on the armadillos or athymic mouse, more commonly the armadillos. That's what you'll see on your board exam. Pathogenic and epidemiologic studies are difficult. Diagnosis is made through histopathological examination of skin lesions. There's the armadillo showing off his feet. Armadillo and then human feet on the right. Another name for Mycobacterium leprae is Hansen's bacillus. Hansen's bacillus. And the disease of leprosy is called Hansen's disease. Make sure you know that, okay? Hansen's disease is leprosy, and Hansen's bacillus is Mycobacterium leprae, okay? It's an obligate intracellular parasite in man. It's confined largely to skin and cold surfaces, especially convex and exposed surfaces, testes, and peripheral nerves. Mycobacterium leprae probably does not grow in internal organisms, and there's two forms of this organism. The first one is lepromatous form. This is the one where tissue samples will show abundant bacilli, abundant AFB acid-fast bacilli on the smear. That's the lepromatous form. And then the tuberculoid form and neural lesions, the organisms are rare and you won't see too many positive acid-fast bacilli. So this is the lepromatous form. A lot of bacilli seen. The lepromatous form, that's what AFB looks like. So when you're looking at an AFB smear, you are looking for anything red. Looks like there's a lot of red here, okay? You can see it, some smears you'll see, I mean, even this much, like 5 on the whole smear. If you see 5 or a few here and a few there, that's positive. If you see 2, only 2, then you have to request another sample. 1 or 2, you have to request another sample and then redo the procedure of digestion and decontamination. Leprosy, the organism, leprosy again, leprosy, and leprosy again. Okay. Mycobacterium fortuitum. This is a rapid growing mycobacteria. Associated diseases are postoperative infection, skin and soft tissue infection, pulmonary infection. Okay, so that's Mycobacterium. The next organism is Nocardia. And the reason why Nocardia is in this lecture is because it's partially acid fast. If you get, and that's a key word that you need to know and learn to recognize. Because on your board exam, if you see the words partially acid fast, right away you need to think Nocardia, okay? Partially acid fast, don't think, well, it's probably one of those Mycobacterium that didn't take up stain. No, it doesn't work that way. If it's gonna take up stain, it's gonna take up stain. But for Nocardia, it's partially acid fast. That's a specific term that you need to remember and make sure that bells and whistles go off. That's not a typical Mycobacterium, it's Nocardia, okay? And the main organism of Nocardia is Nocardia asteroides. Nocardia asteroides. It's gram positive, has a beaded appearance, variably acid fast, catalase positive, and it's aerobic. So it's partially acid fast. Nocardia, asteroides, associated diseases is immunocompromised. These organisms like to wake up the organisms in an immunocompromised patient. Invasive pulmonary infection, disseminated infections. Immunoincompetent, the infection is mycetoma. So this is nocardia now, okay? Lymphocutaneous infection, skin abscesses, and cellulitis. Specimen requirements. So Mycobacterium is recovered from a variety of clinical specimens, and the greatest single problem in recovering Mycobacteria is the presence of large numbers of contaminating organisms, like bacteria, like funguses, organisms that are not acid fast. So the procedure, the isolation procedure, or the preparation procedure includes the elimination of all these bacteria and or funguses, okay? And it requires the procedure called decontamination and digestion. So you get your fresh specimen, morning specimens for sputums, process it immediately. And if you're not going to process it immediately, make sure you refrigerate it immediately within 1 hour. You need to make a decision. Are you going to process it in? How long will it take for you to get to the specimens for processing? If there is no normal flora, digestion or decontamination are not required. For example, CSF does not have normal flora, so you don't digest or decontaminate. Same thing for urine. There's no normal flora, digestion and decontamination is not required. Sputum, early morning specimen, 5 to 10 mLs of deep sputum, and 3 specimens collected on successive days, 8 hours apart. Okay, so you want 3 specimens each day, then you're gonna collect it all, or the lab's gonna pull it together, and they'll do the decontamination and digestion from there. Patients with a non-productive cough, so the patient can't cough, what they'll do is they'll induce the cough expectorant using 10% sodium chloride, okay, and they will force the patient to cough up the sputum. Or they could use ultrasonic nebulizers, are also very effective. A gastric lavage, that's when they put a tube down your esophagus and they take gastric contents. That's called a gastric lavage. And you can get these from infants or others unable to cooperate with sputum induction. So if you can't, if the sodium chloride doesn't work, then they'll just go ahead and go into your stomach and do a gastric lavage. For persons who swallow sputum, you will get the gastric lavage, and the collection bottle should contain alkaline buffer. Okay, so it's already getting the specimen in preparation for the decontamination and digestion. Urine, it's a urine sample, early morning, cleanly voided, midstream portion. Multiple specimens is better than a 24-hour pooled specimen. So multiple specimens of clean catch, voided, etc., is better than a 24-hour pooled specimen. Okay. Other specimens like joint fluid, spinal fluid, pleural fluid, exudates from the lung, and actual tissues. Large quantities are preferable. Okay, the bigger the sample size, like more volume or more sputum or more tissue, the better. Okay, the better your recovery. Make sure you dilute your fluids with saline or buffer, and that improves sedimentation. CSF, you centrifuge for 30 minutes at 3,600 Gs. And you use the sediment. Once you spin it down, you use the sediment for smear and culture. Okay, so again, you're not gonna digest and decontaminate. Homogenization and decontamination. Usually homogenization, you can use that for tissues. If you have tissue, there's a thing called a homogenizer, and it actually pulverizes the tissue. but the homogenization and decontamination are required for sputum, gastric lavage, tissue following grinding, and other body fluids except urine, and then stool. That is actually a common to stool for AFB. Decontamination and contamination processing, you want to do this underneath the hood, biological safety cabinet. And the reagents, make sure you know the reagents, the sodium hydroxide, the decontaminants are sodium hydroxide and zephyran trisphosphate. Sodium hydroxide and zephyran trisphosphate. Those are the decontaminants. And the digestant, make sure you know this, is N-acetyl L-cysteine, NALC. The digestant is NALC, N-acetyl L-cysteine. This will liquefy and turn mucus into a liquid consistency. And then there's the neutralization solution, and that's basically a buffer. Okay, that'll maintain pH, a phosphate buffer. Okay, so decontaminants, digestant, and neutralization solution. Neutralization is filled the tube with phosphate buffer, and the pH is 6.8 and within 1 to 2 centimeters from the top. So you're just adding that much buffer to the specimen. Concentration, that's the centrifugation portion, and you seal it in, you centrifuge it in a sealed container. You can't just put it in the same centrifuge that you're spinning down blood. Usually this type of, the buckets are the type of buckets that have the caps that you screw on to the actual bucket. And then when you centrifuge it, it has to be in an enclosed space. You don't want to centrifuge it where any aerosols that come from the centrifuge can actually infect you. So where I did my training and we processed AFBs, the centrifuges were refrigerated centrifuges, and they were located actually in kind of like a closet, not a closet where you see clothes, but a very, very tiny room that only the centrifuge can fit in there. But the reason is because you don't want to centrifuge and expose yourself to any aerosols leaking from the centrifuge. All right, recommend a refrigerated centrifuge and to guard against aerosols. Specimen processing, you want to incubate. Once you finish your digestion and concentration, you're going to incubate the specimen onto your media. And like I said, typically it's the Middlebrook or the Lowenstein-Jensen. Okay, so these tubes are placed in 35, 37 degrees, 5 to 10% CO2. So if you suspect Mycobacterium marinum or Mycobacterium ulcerans, then you don't want to incubate it at 35, you want to incubate it at room temperature, 25 to 30 degrees. Make sure you know this. AFB cultures are kept for 8 weeks, 2 months, okay? AFB cultures are kept for 8 weeks. Fungal cultures are kept for 4, okay? And each day, and usually in the microbiology lab, it's one of the last things that you do because they're really easy to read. All you do is pull out the tube, look at it, look for growth, no growth, no growth, put it back and do the next pair, okay? It's not very common to see positive cultures, so it's one of the easier benches to read. Growth, no growth, and if there's growth, what kind of growth? And you're looking for the breadcrumbs for tuberculosis, all right? MTB, very infectious, can penetrate the skin, is very resistant to killing, and it will survive 5% phenol for 24 hours, and it's very resistant to drying. A biological safety laboratory 2 is required for those labs that process Mycobacterium, but if you're culturing MTB and M. bovis, you now have to upgrade to a biological safety laboratory 3, from BSL 3. Hospital workers that are tuberculin negative laboratory, that means that your tuberculin or your PPD is negative. Your PPD tests need to be done annually. You want to avoid aerosolization, being around people who cough a lot and or direct contact. All procedures and work performed in a laminar flow hood, class 2A or 2B, with ultraviolet light. The UV light, the ultraviolet light, is there to kill the organism. Okay. Now, when you're not using the hood, that's when you turn the UV light on. Obviously, you don't want to expose yourself to the UV light. So when you're underneath the hood processing specimens, make sure it's off because it can be harmful. For PPE, typical PPE like gloves, lab coat, but you need to include a mask. Centrifuges and shaking machines, these can generate aerosols designed with safety features. Keep them inside the laminar flow hood, the centrifuge and the shaking machines. They need to be underneath the hood. Avoid aerosolization of the inoculum when using inoculating loops. For example, if you're using that's why we're using the plastic disposable. The incinerator, the ones when you use the metal loops and you stick it in the loop incinerator, that can create aerosolization. So that's why I like using the plastic disposable. Splash proof your discard containers. So properly discard of contaminated fluids when you're done using a specimen. Partially fill. You want to add 5% phenol to those specimens that you're discarding. Make sure you use germicides and disinfectants. It must be the germicides and disinfectants must say on the label that it's tuberculocidal. That means it'll kill MTB. Okay. Acid-fast staining procedure designed for organisms with high lipid content. We know that by definition, that for being acid-fast, capable of binding the fusion dye, and it's usually carbyl fusion, that is not removed by the acid alcohol. That's the fastness of the organism, the quote-unquote fastness. Cell wall will retain the stain after decolorizing with acid alcohol. The cell wall will retain the stain after decolorizing with the acid alcohol. And the acid alcohol, like I said, consists of concentrated hydrochloric acid. So the stain will confirm acid-fast nature of the organism. Monitor patients on antimycobacterial drugs, and the staining will help determine appropriate dilution for susceptibility testing. And susceptibility testing is not, it's not like you can do a Kirby Bauer where it gets your growth on Mueller-Hinton aggregates, and you're not going to wait 8 weeks for growth to appear. So what it is the organism will grow in broth media, and then you add your antibiotic disc, and then you look for growth, no growth. So that's how you do it for anaerobes as well. 3 reagents of the stain. Okay, the first one, or 3 types of stains. First one is the Ziehl-Neelsen stain and the Kenyon stain. Those 2 staining procedures use the same components: the carbyl fusion, the acid alcohol, and the methylene blue. The Ziehl-Neelsen stain is a hot stain where you're going to be passing a flame underneath the slide when on the staining rack, and the Kenyon stain does not use flame. The third staining procedure is the fluorochrome stain, and that utilizes the ultraviolet microscope. The first component is carbyl fusion stain. That's how it's made up. But that's kind of like your crystal violet. It's your actual stain that the organism will take up. You put it on the slide, and then depending on whether or not you're using the Kenyon or the Ziehl-Neelsen, you add heat to it. The next reagent is acid alcohol. Like I said, concentrated HCl. When making it, you want it added slowly to ethanol, and solution can get hot or warm. And the third is the counter stain, kind of like safranin, and the counter stain is methylene blue. The fluorochrome stain uses auramine rhodamine reagent. So kind of remember that reagent, auramine rhodamine, and that's the reagent for the fluorochrome stain. Okay, auramine rhodamine reagent uses acid alcohol, And the counter stain is potassium permanganate. Okay, it's not methylene blue. And it requires the use of an ultraviolet microscope. Okay, the Ziehl-Neelsen stain and the Kenyon stain techniques are theoretically the same. It's just that the former is more sensitive in detecting lightly staining organisms. So the Ziehl-Neelsen is you make your smear, you take your smear that you're going to look at, and the heat fixed dried smear, and what you do is you apply 5 to 7 drops of carbofusion stain. Okay, so your smear is dried, and then once it's dried and ready to stain, then you add 5 to 7 drops of carbofusion. Basically, you're putting stain on your dried suspension, and make sure you cover the whole area where the specimen is at. You thoroughly cover the smear. For the Ziehl-Neelsen, just remember that the Ziehl-Neelsen is the hot stain. Ziehl-Neelsen is a hot stain. Kenyon is the cold stain. Kenyon, cold, same hard consonant sound. Kenyon, cold. Ziehl-Neelsen is hot. So, but they both use carbofusion. So in the Ziehl-Neelsen, you have carbofusion on your smear. And when it's on your rack, you take a flame, and we'll probably use wooden applicator sticks. You'll get a flame going, and then you'll pass the flame underneath the smear until you see steam. Okay, you don't want to cook this thing until you see bubbling, and then it actually ends up drying. But you want to heat the slide until you see steam coming off of the carbofusion. Don't allow it to dry, and use the flame or heating rack. The Kenyon stain does not utilize heat. So what you do is you take your carbofusion, and then you allow it to stand for 5 minutes, and that's all you do. Let the stain work into the specimen, and wait 5 minutes. No heat. You do not steam. And then you rinse the slide with the acid alcohol. Decolorize with acid alcohol. No more stain appears in the washing. It's 2 minutes exposure with acid alcohol. And then finally, you would counter stain with methylene blue, 1 to 2 minutes. So you would apply, or you would add enough methylene blue to add to the specimen, and wait 1 to 2 minutes. Rinse, drain, and air dry, and examine under 100x oil immersion oil objective. Okay. You see red, that's a positive AFB smear. Mycobacteria stained red, because your decontamination and digestion procedures should have gotten rid of all the other organisms that you don't need to be looking at. So Mycobacteria, you see any red rods, then that's considered a positive smear. The background will be light blue. Okay, so this is positive. You can barely see it, but you can see that there's red within the cells or around the cells. Here's another one smear where you can actually see the red here. It's not so clear here, but you have to look for it, but it's actually numerous. So these are 2 examples of a positive AFB smear. Definite positive here and definite positive here. Precautions. Observe safety when working with flammable materials. You don't want to burn down the laboratory because you're handling live flame. Follow recommended safety precautions in handling Mycobacteria organisms. Do not allow the slides to dry prior to washing. QC, positive control slides prepared in advance. So we're going to be using positive control slides as positive smears, and we're going to go through the staining procedure. Positive control smear and a negative control smear, make sure you compare the 2. Anything red, small, slightly curved bacilli is considered a positive test. Possibly beaded and tapered ends, blue background. I use M. tuberculosis strain H37RV as quality control organisms. Negative smear review should display numerous blue non-acid-fast bacilli. QC of decontamination and concentration procedures. Okay, reporting acid-fast bacilli. You need to examine the smears carefully. And the rule is you need to look at a minimum of 300 fields. 300 fields before reporting a negative sample. So you need to thoroughly review the smear and look for anything red. Acid-fast bacilli appearance, purple to red, slightly curved. You see any red, it's a positive smear. Non-tuberculosis species such as Mycobacterium avium complex appear pleomorphic, usually coccoid, but it's going to be red still. If large numbers of typical forms of acid-fast bacilli seen, it's reasonable to assume that they're Mycobacterium tuberculosis. Reporting. If you see red rods, positive for acid-fast bacilli, and then if you don't, you call it negative. If you see only 1 or 2 rods on the whole smear, then it's recommended that you do confirmation with a new specimen. But if you see only 1 or 2 organisms on the whole smear, that can't be called a positive. You need to request a new organism and confirm prior to reporting. And this is the reporting table based on the number of organisms you see per how many fields. So 1 to 2 per 300 fields is questionable. So you have to look at 300 fields. That's the minimum. You want to see 1 to 2, that's questionable, request another specimen. If you see 1 to 9, so it's greater than 2, then you call it 1 plus. 1 to 9 per 100 fields. But if you see 1 to 9 per 10 fields, that's 2 plus. And then, or 1 to 9 per field, that's 3 plus. Or greater than 9 per field, that's a lot, and that's 4 plus. Okay, I can't remember if I'm going to be asking how to grade positive smears on a quiz or an exam. But that's AFB, that's the acid-fast bacillus lecture. Are there any questions? Any questions on AFB? Okay.